Um, so, yeah, good morning, first of all. Yeah, I should probably start giving a bit of the background, how we started off on this. I'm certainly not a biologist, and I'm an electrical engineer by education, and we do like communications, coding theory, information theory to some extent, and physical layer security. So this is like starting as a detour, and our university is relatively small. I think we have something like 1,500 students. So colleagues know each other. So you're sitting together at the lunch table. You're sitting together by accident with your life science colleague, and you start chatting. And then it's like, actually, what you're doing is pretty related to our stuff. And then, luckily, our German counterpart of NSF, which is called DFG, had a framework project which liked to bring together biologists, life scientists together with engineers. And then at the first call we tried, and we realized, okay, we have to learn too much from each other. And we said, okay, we are not making this for the first call. We try 2 years later. And then indeed I started like at least half a year before, reading life science papers over weekends to at least be able to talk to each other. And I'm 100% sure I forgot a lot, and I have lots of gaps, so don't expect that I know all the biology terms. I think I will not. And especially the trick is you read the papers from the other discipline and you ignore all the terms. You will never remember what is the name of that amino acid and protein or whatever. Forget about it. This is my viewpoint. Just read to the end of the paper and tell you, this is the idea of the whole thing. And I don't need the names. Forget about the names. Engineers are very bad in remembering names by heart, so it's hopeless. So this started off with Georgi. This is my life science, or was my life science colleague at our university. Now he's back in Georgia, not the Georgia in the US, the one in Europe. And he had a postdoc, Patrick, and Patrick was, essentially or bioinformatics guy, I should say. And David was my PhD student and later postdoc. So I put the young guys in front for the good reason that, well, we invented the ideas, we came with the ideas, but the heavy work had to be done by them, as often the case. Okay, this is the background of the story, and we tried to understand. We didn't like to do really communications. It's not molecular communication that we're interested. We tried to use our tools to give a bit of understanding what the structure of the DNA is, what the levels of protection priorities is in the DNA, and we are sure we did not get the whole picture, of course. In the last response to our proposal, actually one of the reviewers said, well, we would be happy with only a little part of what you are promising. You will find this is already a big step forward, and I hope you will see some of the steps today. This is a bit the structure. So I will start off trying to understand why the mapping between codons, so like 3 base pairs, and amino acids is like the one that is given in that famous table. And this is a strange structure, and you like to understand why is it so, and you like to describe also the mutations as a channel. So people in communications have channel models in place, and as soon as you have a channel model, you can apply information theory and whatever to get a bit of understanding. And then you find a term, set partitioning. This is something that is used, for example, in modems. We talked about modems. There are schemes that partition a signal set into subsets and protect subsets. And this idea we try to move into communications and see if we can argue why certain synonymous mappings are there and at other places they are not. Then the question is, this was actually our starting point with my life science colleague. We try to understand the relation between our information theoretic terms and stability in the DNA. There's, of course, GC content that is very known to biologists. The GC content is somehow more stable than AT and so on. And this goes over also from origin to terminus in the at the bacteria. There's a certain trend over it. All these things are known, also lagging and leading strand differences. All these things are known somehow, but we like to get a more information theoretic picture of it, linking to thermodynamic stability. Then we had results on essential and non-essential genes. We asked the question like, can we see that just from looking at the DNA? Can we classify them from learning from past classifications? And at the end, I will, of course, the whole thing about today and tomorrow is, I think, we try to collect some open questions, and actually there are many. And all these things go through a lot of frustration. I remember that when I did the set partitioning work, I actually promised this for a conference, and as one of these invited talks, and I had not done the work beforehand, and that was a bit risky, and I got it, but it went through some frustration, and I think you know that kind of frustration. Okay, so I start off with codons and amino acids. Why is the mapping? I mean, this picture you know, so I don't need to discuss this. So three codons of 3 base pairs are mapped to an amino acid, and the table is this that you know. And the question is why? So you see there are synonymous ones that map to the same thing, others are not. And then the question, what is the reason for that? And we try to go through a few aspects of that. And the first thing that we have to do is, when you think of communications or information theoretic aspect, you have to think of, okay, I need to describe that channel, that mutations I have to describe as a channel, and we like to do it on a codon-based scheme. So I don't go through all of them. We essentially need a codon mutation probability, and there are models, and this is this ECM model, and we took one by another Schneider, not the one that we just heard beforehand. And there's a so-called ECM matrix. This is the basis for our channel description, and the channel looks then like this. So you see, this is too tiny. I'm sorry for this. So these are all the codons, and the codons on the other side of the channel describes the mutation probabilities from one codon to another. And then the question is, okay, we got this data. The data was from different species, mixed at different times and whatever. So I had contact with the author of the paper, and he told me what is the timing roughly or the intervals that you think of for mutations. And we tried to find out, okay, when we take this model now, these probabilities, and we just change the time scale. And the time scale means you need a power of that matrix that is described by this. And the power of a matrix is actually, when you do singular value decomposition, you do that with the diagonal matrix. In the middle, you take this power, and you look at how does this influence the whole scheme. And you just, as an information theorist, you would describe, well, you have the capacity. Here I would talk about mutual information, which is a step beforehand. I won't be able to derive it. I would also need that hour to do the same step. So I won't do that, but I show you the information content. This is roughly here, with the biological distribution that you actually have between the codons. You describe what this information content, which is the entropy. This would be one of the steps. We saw the log 2 of the probabilities. This is the mean of it. So you would get this as the information content, and then you would describe this model, and you change the exponent, which means you go in time and you see how the capacity of this model, or the mutual information of that model, drops down, and you see there is a crossover point at some point with a certain exponent, where below that you would say, okay, the capacity is not enough to keep that information content, so the species should die, essentially, or change to another one. This is the other option, but that kind of species should actually disappear. And what I mean, of course, we didn't expect an exact number. The underlying data is not even exact. This is also something that one, as an engineer, one has to learn. Biological data that you get from experience are never completely safe and not always reliable, but give an idea. But what's nice about it is not a completely different order that we got. So this means the period that colleague had in mind, that the species would be intact somehow. When we take a power of 0.26, this is not like a factor of 100 or 10 to minus 2 or something. It's very close to 1. So obviously, His data was not out of the world. Our investigation, it was also not. But of course, it's not a totally reliable picture, but it gives you an idea how this would go. So you can actually use capacity to find out what is the timing roughly when a species should not be alive anymore or change. We try to actually find out the actual capacity, the optimum capacity, not the one with the biological distribution, with the so-called Arimoto Blahut or Blehut, however you pronounce it, is the actual capacity. And you see it's pretty close to the actual biological distribution. And when you look at the distribution that you get from this algorithm and compare it to the actual biological one, well, it's a bit different slightly, but you see there are similarities. The Kalbeck-Liebler divergence that you use for differences between distributions, I don't derive this here, is not that great, but when you look at the distribution, it's not so far still. So it's okay, and the difference in capacity is not bad. So it means the biological system was actually kind of able to pick a pretty optimum distribution, pick in quotes. Okay, the next thing, we like to understand that mapping, right? And what the relation is to chemical properties. And we did how should I say? We were somehow a bit less picky in defining a statistical model. We said, okay, everything is Gaussian, and the probabilities that we have between codons, we see it based on a distance, like you would have points in a space, and this distance, and we say it's Gaussian, so the probability to cross over to the other point is the complementary error function, or Q function if you prefer. This is, of course, just a claim. It's not Gaussian, of course, but never mind, we just do it. And we looked only at a pair of 61 times 61 codons, eliminating stopped codons and the like. So what we then did is we did a dimension reduction and tried to plot them in 2 dimensions. You can also do it in 3, but in 3 I would have an animation to do that. So the 2-dimensional plot is much easier to see. So you see all the codons, and you see them represented in 2 dimensions. And you see the corresponding mapping to the amino acids. All these single letters are amino acids, and you see a certain clustering in there. So just by looking essentially at this mutation matrix, from that getting distances, and the distances represented in a 2-dimensional plot gives you locations that you can nicely cluster, and they belong to the same amino acids, which means these are the synonymous ones that are in the circles. And now you try to do the same thing with chemical properties. We took 3, actually, like polarity, size, and so on. And we got these points in 2 dimensions. There's an outlier. One would, probably biologists would know at once why this is an outlier. Don't discuss this, but this here are the corresponding points when you take, I think we took 3 properties for this, and we got them also. And when you would, I would need a second screen now, when I would show both together, then you would see the clustering is alike. I would have to pick some now. I should have picked some. Let's see where PTA is. PTA is here, and S is a bit an outlier here. So PTA is, where was it here? Now I don't see it here together. See some others, HRQ, where's HRQ? Now I have to look myself. HR, where's R? This is here. So you see the neighboring relations are kind of the same. There are some exceptions. There's a list of exceptions. Me as an engineer, I could not argue why they are exceptions, maybe apart from this one, but maybe a life scientist could. So when you would go through the list, you would probably see, yeah, there's a candidate. I understand why this is an outlier. So what is the reason of that thing? So this relation shows you that, first of all, synonymous ones, of course, are mapped to the same amino acid for good reasons, as we'll see. And then when they are close in that sense, the the distance that is produced by the mutations, then you find them also close in the chemical properties. So this means when the distances are very tight here, so the chance to have a mutation to a codon that is synonymous, you will not have any effect. It maps to the same amino acid, so no change at all. When you have a transition between neighboring groupings here, then the chemical properties are still close. This means when you have a mutation to another cluster here, you will get an amino acid that still fulfills the function to some extent, which is nice, right? So the mapping makes a lot of sense. Actually, when you look details, but you need a bit of time, you would see the Taylor classification also popping up. You can do that, but I cannot do that on that size. This is just hopeless. You would not see the components. This is the list of exceptions. I will not go through them, but might be nice for life scientists to look through them and say, yeah, I know. I know. I don't, but you may know. The next thing that I would like to do is looking into coded modulation, what this is, and I show you this one. This is how Ungerberg, a Viennese guy, and his colleague, that is pretty famous in communications. Gottfried came up with this a long time ago. He said, okay, I have actually in parallel IMAI also for so-called multi-level codes that I will use actually more in the next slides. He said, when I transmit on a satellite 8 different points, I could subdivide them in equal partitions in 4 PSK subsets that were on the satellite connections before. They are also still up there because satellites you don't replace all the time, right? So 4 PSK transmissions, or you have 4 points at the same power level. He said, okay, I'm transmitting 8 PSK instead, and I use the redundancy of having more points to protect this. And he did it as subdivisions in that form, and then he constructed a so-called trellis. I don't do that here. It's also not important for our thoughts. So you use that scheme, and when you would now say, actually, when I would have to detect this, then the detection, if I'm in that subset or that one, is really critical because the points are close to each other. When I have to decide between these two, then it's not so critical, right? So the most critical one is here, then less critical here, and then this one is least critical because it's opposite. You might not have to protect it at all. And then you can come with other code constructs. This goes back to IMAI, roughly the same time, and then you actually design codes for the levels, and the codes for the levels to protect these, you have to fulfill a certain rate, and the rate is defined by information theory. And this would mean you have to draw capacity curves. You see the signal to noise ratio here that was just mentioned, and you see they are flattening out at the number according to the number of points. When you have only 4 points, you cannot transmit more than 2 bits. We just heard this. Thanks for the nice introduction. I don't need to do that now. So you see these curves flatten up when the signal noise ratio goes high. They flatten out according to the number of points that you have. And then the spaces between them gives you actually the rate that should be present at for the protection of the different levels. These are the code rates, so the number of information bits divided by the number of code word bits. So you can get this, and then the idea is, okay, can we do that in a similar way also in our application? And then you like to do a partition tree also there. We started at the second base pair in the middle because this defines principal properties, so we did not start at the first one. So we started at the second one, so you see the 4 possible base pairs, and then we took the first and the third. Why the third at the end? All life science colleagues probably know the wobble rule, so the last one is the most shaky one, I put it like this. So we put it in that order, so the second bit first, the second bit, you see, I'm a communications guy, the second base pair first, and then the first, and then the third. You can do the set partitioning tree, and then you can try to define individual capacities for the levels, and then also the differences between them, because the difference would be the code rates in that set partitioning thinking. So these are the properties that you have when you define the second base pair. And there's a bit of information theory. This is the so-called chain rule, but I won't derive that here. This is would take too much time. It's actually the Bayes rule that you know. Bayes rule in information theory terms becomes the chain rule. Bayes rule you know from statistics and probability courses. These are now the capacities, and you see the subdivision this is the first partition, so this means I'm looking at this level here. And maybe I should point out this guy here, which has these lots of synonymous mappings. You see the capacity of this one. This is what is called label PSTA. According to here, PSTA is the label. This goes down to 0 very quickly. So this is the worst of the guys. So no wonder that the partitioning stops, or the subdivision stops early. Okay, this I will point out. One could probably look at more details here, but I'd like to show some others. This is for the second partition. So when you look at the capacities that are happening here at that position, what's interesting there? Let's look at the ones that say go to 0 very quickly. This is more maybe the most visible thing compared to those. These keep capacity for a long time, but this one is going down to 0 very quickly. And when I see the color code, it's this what is labeled with V and L, and this has the first one U. Let me see where U was. U was here, and we had these guys, right? So these were the ones that you had synonymous mappings. So this means when you compute the capacity, you see the ones that drop down very quickly, those are the ones that don't contain capacity anymore, obviously, right? So you cannot do anything with it anymore. So when it's already at the second partition 0, what should you do with the third one? There is no. So this means this argues very quickly why you would actually have the synonymous mappings just from the mutation matrix that we got. You can compute these things and you see which ones should be synonymous mappings, which make a lot of sense. So this is chain rule again. I don't do that. These are the differences now according to this partitioning scheme. And what would you see there? Okay, these are the levels 1, 2, 3, right? So 1 is the dotted line here. C2 is actually the same, right? So these two guys are overlapping. This would be the capacities that you think of error correcting codes that nobody knows, or most don't know where they are, but maybe we find them. When you look there, you see these two are the same. The third one, the lowest level, where you have that wobble rule would hold essentially, where you would have these lots of synonymous mappings. This is very, very small, right? So this is visible. The last one is, according to wobble rule, the bad one, and this is also visible. The other two are the same. Why they are the same, I don't know, but the reasoning might be that during evolution you had a mapping with only 2 base pairs. There was this claim to have a comma function afterwards and things like that. So this might actually be the reason for it. The other aspect is it goes down to 1. I mean, this is 1 bit, right? It's not base pair. It goes to 1 bit. So essentially, instead of 4 possibilities, it goes down to 2. So this means how can you survive with only 2 options? So essentially, this would mean at some point the essential mapping would go down to only 2 possibilities instead of 4. So is this sufficient? How can this survive with only 1 bit content per base pair when you only look at the first 2? Okay, I'll try my best. Okay, thermodynamic stability, I will do that very quickly. So let's just look at a few pictures. I ignore all the theory. I use Gibbs entropy for stability and the Shannon entropy for the other one. Let's just look at some pictures. You will see the Gibbs one, which represents thermodynamic stability, and the Shannon one, which is the blue one. And you see them in windows over the bacterial DNA. And you see, wow, they are pretty related, right? So stability goes in line with the properties that are also given by Shannon's entropy. The changes are not so dramatic, but you see the relations. This goes further. I don't go through all the species. This there is very clear, right? It's very obvious. You don't need to look deeply into it. The other thing is when you classify genes into anabolic, catabolic, and so on, you could also see the relation. So when you have, for example, the red and green one, the green one is Gibbs entropy. I only plotted one because the other one is related to it. You see there is a clear dependency to the red one, and the red one is the first guy. So there's a direct relation. So this means the placement of, or the probability of certain gene positions are in line with the entropy calculations. This is also pretty interesting. And the rougher shape here is because of quantization. There are not so many in there. Okay, another issue here. Essential and non-essential genes. So essential are the one that you need to have a bacterium or whatever it is to survive. And the question is, can you do that? There are many like features that are non-information theory. Down there are some. We are more interested in mutual information, entropy, and so on. The last one is Markov properties. When you think of sequences, we as engineers, for a source that has sequence properties, that has memory, you think of a Markov model as a model for the memory. And you can also apply that here. So we use all these together. We do some machine learning algorithms, which are also listed. I don't do the theory here. I just skip that. Essentially, this was here. We use, for example, these two, support vector machine or random forest. You might take other machine learning algorithms. Doesn't matter so much. The interesting thing is when you combine these properties right, you get actually a classification quality by just essentially reading the sequence. You don't need to go to the wet lab and do experiments. Just from the readings, you get as good quality as when you have to do it the hard way, which is interesting. So you essentially learn from, say, 80% of a DNA piece, you can predict 20% that are left over, or also between species, but the species should be halfway closely related. When they are too distant, it works less well. But this is obvious because the sequence relations are different. But you can do these kind of things. So you can just look at the sequence, have a machine learning algorithm, and predict where the essential genes are located later on. And then in the wet lab, you can check if you want. But it's almost, you do this area, and I don't like to describe that a lot. But when you think of the other possibilities, they are in the same quality domain. So this is interesting. So you can just do it in a relatively easy manner. Okay, I should just show a few pictures to remind you. There's one that you haven't seen, and this is the one here. This is gene regulatory networks. This one is actually one that is for joint lethality. So when you have two genes that are turned off by mutations, then you will also have the species or the bacterium or whatever it is dying. The others you have seen. Open questions are like, what are the outliers here? Why are they like this? What can you interpret more than this? And just looking at it, say, it's related. More we cannot do, but maybe some of you in the audience can. This we have done a lot, and also other authors have worked on this. So I think there is a lot of work being done. This becomes a very tough thing. I mean, those of you that have worked with these regulatory networks, I have seen them before already in earlier times, like, wow, this becomes complicated. And this is probably, when you think of error correcting possibilities, the first one is the mapping. This is now pretty understood how this goes. Then the question is, what is the highest level of error protection? And this goes to regulatory networks. How do you understand them? And I started looking into them, and it becomes a nightmare, I have to admit. When you try to move that to a coding structure, it's not, unfortunately, our nicely linear codes, like LDPC codes or whatever we like to use. Unfortunately, this is very nonlinear. And frankly, when I was trying to move that to a coding structure, to a graph of a code, what should I do with it? So frankly, I still don't know, but these are the next steps to be done. Okay, I should stop, I guess, right? Thank you.
Time for one question and then a break.
This is a reminder.
Yeah. I don't want to. Oh, sorry. Is someone else? Because I already asked a question. So does someone else want to? Yeah, go for it.
Yeah, we need to use the microphone for the video, especially.
So early in your talk, you pointed to you mentioned, I think, the mapping between codons and amino acids, specifically in their chemical properties. You mentioned the volume, and I also saw a mention of hydrophobicity.
Polarity was there. Right now, I forgot the third one. I should remember, but I don't remember right now. So we use third, but you can use more.
Okay.
I think authors use up to 24 or something like that. So you can take more chemical properties and boil them down to that plot in 2 dimensions. I think we tried a few others, but there is work out there to take more. So this figure is not unique in a way. Other authors have done this also with even higher number of properties.
Right. Yeah, there are a lot of properties you could choose. It's kind of my field. I'm just curious, what other, if you saw those patterns with just a specific set or a few properties, or if regardless of the properties you chose, you saw those patterns?
It's not regardless. Also, how you do the I mean, for the chemical properties, of course, it will change a bit when you use more properties. I think we took the 3 that appeared to us the most important. So we didn't like to open the bucket for all of them. We like to see the principal behavior.
Right.
But you certainly can be more exact if you want to do that. So we stopped at that point. We said, okay, we see the relation. We like to actually go a bit further and try to do this mutual information thing and understand why you have that mapping. I think you can be more sophisticated on this. I'm not claiming this is the best you can do.
Okay.
And also here, it depends a lot on your choice. If you take a Gaussian assumption, this was just for communications people, like this is a nice model to have. It's of course often not realistic, but it's so easy, right? And I'm sure it's not Gaussian at that point. Why would a mutation process be Gaussian?
Right.
Probably not. It's hitting by whatever radiation or something. This is not a Gaussian process. It's more something impulsive. You might have a heavy tail distribution or whatever. This is just a very off-the-shelf idea to move it to distances, to have something to plot.
Okay.
But you can certainly do better, and you can also do better clustering. There are many ways of doing. We tried some other clustering algorithms. They got other outliers, so we said, okay, then we can also stick to this one. It was not giving more insights. Okay. Some outliers stayed the same, some others showed up. It depends also a lot how many dimensions you go down to. When you go down to 2, then it's nice to look at it, but in 3 dimensions you can actually see a bit more structure still. Because when you look at the eigenvalues that are in the scheme, they are not in steps down. So you should actually probably take more dimensions, but I can only see something in 2 or 3. Right. And in 3 I cannot do that in talking. I need an animation to draw to rotate this to have an impression somehow.
All right. Yeah, very interesting. Thank you.
Welcome.
Thank you very much. I think we're on a break.
Maybe we can have a quick question from Eric. Actually, it's a related question. So this may be naive, but so in constructing that 2-dimensional plot, or in thinking about this, the channel, it seems, is from one generation to the next, because you're using a mutational matrix. Like, what's the chance of the codon changing from one value to another through mutation? I'm just wondering because one of the other channels that's present here is the actual transcription and translation, right? You're moving from DNA to RNA and then RNA to protein. And people have characterized the errors in that process, just by looking at how often do you get mistranslation. So I was just curious if you've looked at that. Some of these codons...
We know, but we didn't.
Oh, okay, cool. Never mind then.
Thank you.